Professor Cech is a distinguished professor at the University of Colorado. He won the Nobel Prize in Chemistry in 1989 on catalytic RNA. And besides that, he has been conferred the National Medal of Science, Heineken Prize, Canada Garden International Award, Albert Laser Basic Medical Research Award, Golden Plate Award, Hummer Gold Medal, and many other honors. Professor Cech is a role model in the RNA field. His discovery of the catalytic RNA fundamentally changed our understanding of how RNAs work by showing that RNA can, similarly to proteins, undergo biocatalysis. In recent years, Professor Cech has turned his attention to looking at RNA's role as a master regulator in epigenetic silencing. So today, we're really honored to have him give us his lecture. Thank you.
Hello, and greetings from Boulder, Colorado in the United States. At last year's wonderful Global Youth Science event, I spoke about some current events that had to do with RNA, such as the CRISPR genome editing, for which Jennifer Doudna and Emmanuelle Charpentier received the Nobel Prize, and also the coronavirus and the messenger RNA vaccines. So having talked about that more general topic last year, I thought I would talk about some work that is closer to our own research, because I still have a very active research laboratory, and we are working on the complete replication of the very ends of our chromosomes. This topic may be a bit technical in some places, but I will try to give a very general introduction so that those of you who are not cell and molecular biologists should be able to follow most of the work. Now, what are telomeres? They are the very ends of our chromosomes. And here you can see this array of human metaphase chromosomes, and they are decorated with a probe that lights up a particular sequence. And this is the sequence of DNA at the very ends of our chromosomes. And you can see that it is conserved, that there's the same sequence at the end, at the two ends of all of the chromatids of our chromosomes. So the question then is, how are these very ends of the chromosomes maintained? And it turns out that in most of our somatic cells, they are not maintained. And this telomeric DNA shrinks as our cells divide. And when the telomeres get to be short, then the cells stop dividing. And this is a good thing because it's a tumor suppressor mechanism. We don't want the cells in our body to keep growing and growing and dividing when we are adults. There are some exceptions. Stem cells, for example, must continue to divide, but most of the cells in our body are better off if they stop dividing. And so the enzyme that does increase the length of our chromosome ends early in embryogenesis, when of course we do need to keep our cells dividing, and also in our stem cells, is called telomerase. It was discovered by Elizabeth Blackburn and Carol Greider, who won the Nobel Prize for that discovery. And as I will talk about it, they also discovered the RNA subunit, shown here as the purple ribbon. And then my laboratory discovered the necessary protein subunit called telomerase reverse transcriptase. And it was really the combination of the RNA and the protein that allows telomerase to work. And this is a good thing for our stem cells, but unfortunately the telomerase gets reactivated in human cancers, and this makes the cancer cells immortal. It allows them to continue to divide in an uncontrolled way, much to our own detriment. Now, Blackburn and Greider also found this RNA component of telomerase. They found it in a ciliated protozoan called Tetrahymena in freshwater ponds throughout the world. And as you can see at the top diagram of the RNA, that this is a fairly small telomerase RNA. Vertebrates, including humans, have a somewhat larger telomerase RNA shown in the middle, and other species, such as the yeast that is responsible for our bread and our beer, is the organism Saccharomyces cerevisiae, has a very large telomerase RNA. But what is common about all of these telomerase RNAs is that they have this region called the template that determines the sequence of the DNA that is laid down at the ends of our chromosomes. And so how does this enzyme work? Well, here is an animation that I hope is playing. And the blue strand is the RNA template that I was just talking about. The green lines are the active site of the TERT enzyme. And then you can see the red telomeric DNA is being extended by 6 nucleotides and then ratchets back, binds again, and is extended by yet an additional 6 nucleotides. So let me tell you first what I will try to explain in the next 25 minutes. So first of all, that telomerase is the enzyme that extends the G-rich strand of the telomeres, this repeated sequence TTA-GGG in humans, somewhat different in other organisms, that the telomerase, as I already said, has both RNA and protein components, that the discovery of this telomerase reverse transcriptase by Joachim Lingner, a postdoctoral fellow in my own laboratory, was done first in euplotes. And this turned out that this work in this pond animal gave a clue to how cancer works. And that's something we do in biology all the time, is we use an organism that makes it easy to learn something about biology. And then we trust that since everything is related through evolution, that whatever we learn in a lowly pond animal like euplotes has a good chance of being relevant to the human condition. That was certainly true with telomerase. Then I'll show you how we can watch single molecules of telomerase moving around a living cancer cell, an enormously amazing technology that allows us to see how telomerase searches for telomeres. And then I'll finally talk about some very recent work about synthesis of the other strand of the telomere, the C-rich strand, which you can see by its sequence is repeats of a sequence that are, of course, complementary. They match the sequence that is laid down by telomerase. Now, again, here is the artist's drawing of the telomerase enzyme. And now I want to point out in a little bit more detail this template region of the telomerase RNA, and it is making the repeated sequence at the end of the chromosome. You may, if you had sharp eyes, you will notice that this is a little bit different sequence than I talked about for human, which was TTA-GGG, because this is the sequence from Tetrahymena, which is the organism from which Grider and Blackburn originally purified telomerase. Now, the organism that we use to find the protein is this little pond animal called Euplotes ediculatus. And why would we choose this microscopic organism in our search for purification of telomerase? Well, that's because unlike humans that have only 23 times 2 in a diploid cell, 46 chromosomes, Euplotes has 100 million chromosomes, very small ones, per nucleus. And this allows it to have a large amount of telomerase. And as a biochemist, we want to use an organism that exaggerates the telomerase, makes a lot of it to make it easy to find. And then we use the Human Genome Project, and it took many years to find the euploides telomerase reverse transcriptase, and then only a few months to use the Human Genome Project to find the related human version of that protein. Now, again, what does this have to do with cancer? And the answer was not found by us, but was found by two groups, Franklin Wong and Levi Garraway's group, and Horn and co-workers at the German Cancer Center in Heidelberg. Huang et al. Were at the Dana-Farber Cancer Institute in Boston in the United States, Horn et al. In Heidelberg, Germany. And so what they found was that there was a mutation in the regulatory sequence, the so-called promoter of the gene that we discovered, the TERT gene, that was present in multiple cancers, but was not present in the normal tissue next to the tumor tissue. And amazingly, 80% of all melanoma samples independently isolated from around the world had the same single base pair change in the TERT promoter. 80% of the brain cancer called glioblastoma, 70% of bladder cancer, 40% of liver cancer. So very common, the third most common mutation in all of cancer after KRAS and P53 mutations. Interestingly, this mutation is absent in breast and colon cancer and some other common cancers, but these still reactivate the TERT gene, and they do so epigenetically rather than through a genetic mutation. And as I already alluded to, this reactivation of telomerase in cancer is a bad thing to have too much of it, but it has to be very tightly regulated because too little telomerase, an insufficiency of the telomerase enzyme, even if you have just half the normal amount, that causes a variety of diseases that are due to stem cell, where a stem cell process is insufficient. And so you don't want too little telomerase in the stem cells. You don't want too much telomerase because it's one step towards cancer. Now I would like to turn to this question of watching the telomerase enzyme move around the human cell nucleus in real time. And the question that we asked was, how does telomerase, a rare ribonucleoprotein enzyme, there are only some hundreds of copies of it per human cell nucleus, how does it find the very ends of our chromosomes? Because that also is a very rare part of the nuclear milieu. And so this is like looking for a needle in a haystack. How does telomerase find where it needs to go? And this was the work of Jens Schmidt, a postdoctoral fellow in my laboratory. Now, before I talk about Jens's work, I have to tell you a little bit more about the structure of the end of the chromosome. And it's a little bit like the very ends of our shoelaces, where we need to have plastic tips to protect the shoelace so that it doesn't unravel. Similarly, the ends of our chromosomes have a group of proteins attached to them that are protecting or sheltering the end of the chromosome. Most of these were discovered by the laboratory of Tizia de Lange, who is at the Rockefeller University in New York City. And one of them, protection of telomeres number 1, shown in blue here, was discovered by Peter Baumann, a postdoctoral fellow in my laboratory in Boulder about 20 years ago. These proteins were all discovered. And one of the things that these proteins do, in addition to protecting the end of the chromosome, is to recruit the telomerase ribonucleoprotein to the chromosome end. So we have mutations both in the shelterin complex and in the telomerase protein, the TERT that prevent this interaction. And then the telomerase, even though it's enzymatically active, it can't find the end of the chromosome. The telomeres cannot be maintained, and so they shrink with time. Now, the DNA is also very interesting at the end of the chromosome. Most of you probably have read or studied that the DNA is a double helix, and certainly in most of the chromosome, this is true. And at the telomere, these TTA-GGG repeated sequences are mostly double-stranded. But at the very end of the chromosome, of every one of our chromosomes, there is a stretch of about 200 or 300 nucleotides that are single-stranded DNA. And this is always of the red strand, which is the strand that has the G's in it. And so the shelterin complex has to protect both the double-stranded and the single-stranded parts of the telomere, and the telomerase has to find the single-stranded end because that is what it is able to extend. So what Jan Schmidt did to mark the ends of the chromosomes in a living cell was to use CRISPR genome editing to put a fluorescent tag on one of those telomere binding proteins, one of those shelterin components called telomere repeat factor 2. And so he decorated all the ends of the chromosomes with one color of a fluorescent dye. He decorated all of the TERT molecules, which are the telomerase, with another color. And he used yet a third color, a blue color, to mark the Cajal body, which is a non-enveloped compartment within the nucleus, which is involved in ribonucleoprotein maturation. And so in this movie that I'm going to show, you will all think that I have sped it up, but in fact, this is real time. I have not sped this up. Molecules are actually moving this quickly, diffusing this quickly within the human cell nucleus. I think you can see sort of the edge of the nucleus here. This particular one is not quite round. It's a little bit elliptical. And the green dots are the telomerase molecules moving very quickly. The red dots are the chromosome ends, the telomeres, not moving at all on this 15-second movie. They diffuse only very slowly. And then again, the Cajal bodies here and here are shown in blue. So now you will see the telomerase moving around the nucleus, looking for telomeres, sort of like angry bees in a bottle. Now, I'm going to play this again, and I want you to watch the place that I'm going to show you right here, which looks like it's yellow. So this is where a green telomerase is overlapping with a red telomere, and you can see that they stay together for the entire length of this movie. And these are the long static interactions that are long enough for the enzyme to actually do its job and extend the telomere. So the model that we can, so of course you can't just look at the pictures to understand how telomerase finds the telomere. You have to track thousands of particles in movies to see where they're going, to see how often they find the telomere. We also use genetics, and we determined mutations that disrupted this activity so that we could make sure that we were seeing authentic binding events because the mutations prevented those events. And then we also used drugs which prevent the telomerase from pairing with the telomeric single-stranded extension. And it was by doing many, many different kinds of experiments that we came up with this understanding of how human telomerase finds the telomere. So we found that it engages with the telomere transiently, short interactions, maybe 1,000 times during the DNA synthetic phase of the cell cycle. So huge number, and that's because it's binding through just protein-protein interactions and has not yet found the base pairing interaction with the very end of the chromosome. So it binds, but it's not very stable, and it is released. And then occasionally, much more rare, it has both this protein-protein interaction, which tells it that it's close to the chromosome end, and if it's right at the chromosome end, the base pairing between its RNA template and the single-stranded DNA, and only then is it able to lock down and actually extend the chromosome end. So in the last part of this talk, I'm going to talk about the most recent work, which has to do with a different protein complex, not shelterin, which I've already talked about, not telomerase, which I've already talked about, but instead this CST, which is an abbreviation for the three genes that make the three proteins shown that make this complex. And it is responsible for stopping the telomerase from continuing to make telomeric DNA. And then it brings in polymerase alpha, DNA polymerase alpha, and primase, which makes an Okazaki fragment for lagging strand DNA synthesis. And this helps the complementary strand, which we call the C strand, to be made at the chromosome end. And Carolyn Price, who's a professor at the University of Cincinnati, did much of the pioneering work on this system. So I had a extremely talented postdoctoral fellow from Singapore. His name was CJ Lim, and he succeeded in doing what had been very difficult for many years in the field, which was to determine the molecular structure of this CST complex, which is an accessory factor for polymerase alpha. Now, initially, he was able to get the structure to 6 angstroms resolution. And you will remember that an atomic bond in chemistry is about an angstrom unit across. So 6 angstroms is not good enough to really see individual atoms or amino acids in a protein, but it was good enough for us to dock into this structure some previously determined crystal structures of little pieces of CST. And it was then that we knew that we were at least on the right track. So what do you do if you are stuck in a structural biology project and cannot get the really beautiful resolution that you would like? Well, the trick, as my friend Tom Stites always used to tell me when we were skiing together, he would say, you always add the relevant partners of the protein. Because even if that makes it larger, which you might think would make it harder to solve the structure, it makes it perhaps more locked down and uniform in structure, less dynamic. And so what partner did we add? We added single-stranded telomeric DNA, TTAGGG repeats. And we had a surprise. Not only was the structure now much more solid and much easier to solve by cryo-electron microscopy, but it self-assembled into this enormous decamer, 10 subunits. Here's a side view. Here's a view from the top. And this superstructure is something that had not been anticipated, had not been predicted, and we still do not know what function the superstructure, the decamer, has, and what functions the smaller unit, for example, this one shown in cyan, which is a single CST heterotrimer, what functions it has. But we were able, now the resolution was so good that you can see that the electron density, which is this gray web, could easily be fit with the amino acids. We can see the amino acids. This one must be one that has a long side chain. This turned out to be an arginine. Here's one that is flat. That turns out to be an aromatic ring of a tyrosine. So C.J. Lim was able to fit in de novo, which means without any cheating, without any using previous structures, 70% of the CTC1 structure that had never been determined before. And he was also able to find that a part of the telomeric DNA was very well positioned within the protein complex. And so only 4 nucleotides were really rigidly oriented. The rest of the DNA was more flexible, and we could not trace it. So that's the structure of CST. Now, how about its function? So what we've been doing recently in the laboratory is to look at the ability of CST to coordinate polymerase alpha and primase to make this C strand. And in this particular gel electrophoresis, we are, first of all, showing you the telomerase reaction products, the TTA-GGG, in this case, just as a way of getting oriented to find out what size is running at each position. So this beautiful ladder of telomerase products was what Blackburn and Greider originally saw with tetrahymena, but this is the human telomerase. So in this case, this is just serving to get you oriented for what size products we're seeing. And then these dark bands and these dark bands that are increasing as we do longer reaction times are the C-strand products that are being made on the telomeric DNA as a template. And so this is what the polymerase alpha primase is able to make. And somewhat like the telomerase ladder, it also has a 6 nucleotide repeat, a little bit less specific than the telomerase one, but nonetheless quite apparent. And this quantification just shows you that the synthesis is linear for a period of the 2 hours that we ran the reaction for. So why is there this 6 nucleotide ladder of C-strand products? Well, this gray cartoon is the telomere single-stranded DNA, and it is the repeats of this TTA-GGG. The sequence is backwards because I'm going 5' to 3' from right to left in this case. And each of these gray boxes represents one of these telomeric repeats. So as a template, we are using multiple telomeric repeats. Now, if the CST positions polyalpha primase on the left end, then it will make a product shown in green that is of length 16 nucleotides shorter than the length of the template. But every set of repeats is an equal target for this CST polyalpha primase. So what if it sits in the purple position instead? Well, then it'll make a product that is 6 nucleotides shorter. It's N-22. And what if it sits in the position shown in blue? Then it will make one that is yet 6 nucleotides shorter, etc. It can start at any of these positions, and that's how we get the 6 nucleotide ladder. And we're very excited, we've been able to get the complete telomere DNA replication by putting everything together in the same test tube. We can put telomerase together with CST Pol alpha primase, and we get both G-strand and C-strand synthesis. We are radioactively labeling here only the C-strand, but we know that the G-strand must be there because if we don't have the telomerase there, we don't get any of these products being made. So the telomerase makes the G strand, and then the CST Pol alpha primase makes the C strand. And we have a number of controls that convince us that these are authentic products. And what does this 0 minutes, 10 minutes, 30 minutes mean? Well, 0 minutes means that we mix everything together right at the beginning, and we do get products formed after 1 hour. But if we give the telomerase a 10-minute head start, or a 30-minute or a 60-minute head start, we get more product being formed. And so the reaction works a little bit better if we give the telomerase some time to make the template before we add the CST Pol alpha primase. And we think that by studying this reaction in the test tube, we can understand the atomic details of everything that is happening for telomere replication to occur. Our current model is shown here in a cartoon form, that the telomerase has this orange RNA template. It uses a primer of telomeric DNA. You can think of this as the end of a human chromosome, but in our reactions, it is a synthetic oligonucleotide containing 3 telomeric repeats. Telomerase makes a long copy of that, multiple telomeric repeats, and then CST Pol alpha primase uses that as a template and makes the C strand, first laying down an RNA primer, then extending the RNA primer, then switching over to the Pol alpha subunit, which makes the C strand DNA, and it can go all the way to the end of the template. So, in conclusion, what I've tried to explain today, and I hope that I was at least somewhat successful, is that telomerase is the enzyme that extends the telomeric overhang at the ends of each of our chromosomes. That it is an RNA plus a protein. The protein is called telomerase reverse transcriptase. That the discovery of TERT by Joachim Lingner in Boulder, Colorado, about 25 years ago, was done first in euploides, but we quickly were able to jump to the human version, and that gave us a clue about how telomerase is reactivated in cancer cells. We can watch single molecules of telomerase search for telomeres, and this has allowed us to understand the recruitment of telomerase to this very rare end of the chromosome. And finally, we've determined the structure of the CST complex and seen through enzymatic assays how it can coordinate Pol alpha primase to synthesize the C strand of the telomere. And what is the future of this field? Well, first of all, we would like to inhibit cancer metastasis by inhibiting telomerase activity, or perhaps inhibiting the CST complex, which is required for the C strand synthesis, or perhaps inhibiting the recruitment of telomerase to the chromosome ends. And then on the other hand, if we have diseases of stem cell insufficiency, such as dyskeratosis congenita, we would prefer to upregulate telomerase so that we could better maintain stem cell activity. Here is my laboratory on a hike that we take every summer, and you can see that we enjoy a variety of people from many different countries and many different backgrounds working together on these problems. And in particular today, I would like to thank Lingner, Schmidt, and Lim, who are all now have left my lab and have their own laboratories. They're professors throughout the world, and also Debbie Woodke, my colleague at CU Boulder, Art Zog, who did the most recent experiments on the CST Pol alpha primase, is in my laboratory, and the Howard Hughes Medical Institute for funding. And thank you for your attention.
Thank you, Tom, for the wonderful talk. So we have some questions. The first question from Bing Lu. Hi, Professor Cech. May I know your opinion about the potential use of telomerase to prolong lifespan and rejuvenation? Thanks.
Well, first of all, I am pleased to be here, and I send my greetings to all of you around the world. I've had a very nice time talking to a group of students and young scientists, and I would like to know whether you can hear me adequately. Can you hear me okay?
Yes.
So I think that telomerase activation in older human beings certainly could be used to prolong the proliferative lifespan of certain types of cells, such as stem cells, if they are not dividing very well because of the lack of telomerase. However, it could be a very dangerous sort of therapy in general because it is one step towards cancer. And so rampant reactivation of telomerase in adult humans would be a bad thing. And we know this because there are genetic diseases where telomerase is active, exceptionally active, because of a promoter mutation in humans, and they get multiple cancers.
Thank you. Another question from Josephine Wong. What are the challenges for using telomerase targeting therapies in cancer?
Challenges for using telomerase
targeting therapies in cancer.
Well, thus far, it has been difficult to make a small molecule inhibitor that works very effectively against telomerase. Perhaps because telomerase is such an intrinsically slow and inefficient enzyme, it doesn't need to work very well in order to maintain telomeres, and so you would need a very good drug to shut it down. The other challenges are the possible side effects by inhibiting the growth of stem cells if you inhibit telomerase, because there are some tissues in the adult body, such as stem cells, where continued proliferation is required.
Another question by Jennifer Love. With the discovery of CRISPR and mRNA vaccines, it certainly is an exciting age for RNA biology. What do you believe are the major next steps for both basic and applied RNA research?
Well, thank you for that question. Of course, I love RNA, so I am pleased to talk about this. I think that in terms of basic research, the big area for the future are the long non-coding RNAs. They outnumber the messenger RNAs in human cells by a large amount. They are extremely tissue specific, so they are different long non-coding RNAs produced in the brain, in the eye, in the skin, in the liver, and so that makes them very interesting, this tissue specificity. And their functions are, to be honest, quite unknown. Even the most heavily studied ones the functions are still controversial, but a number of them are essential, and there's good reason to believe that they are doing something important. So that's the big area for the future. There may be more than 100,000 of these long non-coding RNAs encoded by the human genome. In terms of the applied research, I think the big question right now is this stabilized messenger RNA used by both Moderna and BioNTech, Pfizer for the vaccines. How general will that be as a therapeutic? I think that it will be useful for additional vaccines, although whether it will completely take over the vaccine world is yet to be determined. But how about beyond that? And the most exciting possibility might be that monoclonal antibodies, which are currently produced as pharmaceuticals with Chinese hamster cells in big fermenters, that perhaps instead of producing the monoclonal antibodies as a drug, one could inject a messenger RNA encoding the monoclonal antibody, and that might be very effective in some cases. But we just don't know. It's going to take the next 10 years to figure that out.
Yeah, that's really fascinating. There's another question from Sumima Singh. What is your advice to young scientists who want to make breakthrough discoveries? Can one do that in the industry? Will one's ideas discoveries remain their own?
Yes, well, certainly industry is a good place to make discoveries. There tends to be a lot of teamwork in industry, which I think is very healthy, people working together towards a common goal. There's more freedom to make discoveries in academic laboratories, where there's not the pressure of having to produce a product which can be sold to keep the company in business. But my students and postdoctoral fellows have really gone in both directions, both in academia and also into industry. And I think there are opportunities for creativity in both cases.
Thank you very much. So there's another question about animal models. So would it be possible to use a model such as the naked mole rat to further understand how to discover and utilize the potential applications of telomeres?
Yes. Some of the, I'm not, I think I would know a naked mole rat if I saw one, but I do not know much about its telomere biology. The more common rodent that is studied, of course, is the mouse, Mus musculus, and its telomere biology is actually quite distinct from that of humans. That the laboratory mice have extremely long telomeres. And so this is not considered to have the cancer relationship that it does in humans. Perhaps unnecessary because mice don't live very long, and cancer is, to some extent, a disease of older organisms. And so right now, much of our work on telomerase is being done in human tissue culture cells, which we think, at least for very fundamental processes, is a good starting point.
We also have two questions that are both related to specificity of targeting of telomerase, and whether there could be a way to target basically telomerase specifically without the side effects of disrupting other cells like stem cells.
Yes, well, of course, one way to specifically target it to cancer cells might be to attach the drug to something that will target it to cancer cells only. And there are very exciting advances in the area of linking antibodies to drugs and making an antibody drug conjugate. Then if the antibody could recognize the tumor cell and deliver an anti-telomerase drug, this might be a possibility. So I think the future holds some opportunities.
Thank you so much. I think in the interest of time, we'll have one last question. So in what way would you expect modern AI methods to come into play in RNA research field?
Well, I just happen to have an article here from Science Magazine written by a friend of mine, Riju Das, Stanford University, with his collaborator Dror. And the title of this article, it's featured right on the cover of the magazine. It's not very old. It is from August of 2021, so 6 months ago, is called Artificial Intelligence Reveals RNA Structure. So I think the future in this area is bright, and I encourage you to look up this article. And that was pure luck that I happened to have that sitting right next to me.
Thank you so much, Tom, for answering all the questions and for your wonderful talk. And thanks everyone for participating. Thank you.
Take care.